The joy of learning biology, right here online for you and me. Explore the wonder.
Dihybrid inheritance. This is the simultaneous inheritance of two characters. Mendel used P shape and P cotyledon color as the characteristics. Crossed pure breeding, homozygous plants having round and yellow peas with pure breeding plants having wrinkled and green peas. BioClass Hub. Let us carry out a cross using two characters. Let capital R represent the allele for round seed. Let small r represents wrinkled seed. Let capital Y represent the yellow seed. Let small y represent green seed. Parental phenotypes, pure breeding round, and yellow seed crossed with pure breeding wrinkled and green seed. Parental genotypes, capital R, R, capital Y, Y, crossed with small r, R, small y, Y. Meiosis leads to the formation of gametes. Any one of a pair of characteristics from this combination may combine with either one of another pair from this combination. It goes like this to form the gametes. This is known as the principle of independent assortment. It forms the second law of genetics. It's also called Mendel's second law. It states that any one of a pair of characteristics may combine with either one of another pair. When we observe that all these gametes are the same, we can summarize it by using a single gamete, the same principle for this side. Random fertilization to form the F1 genotypes. F1 physical appearance, all round and yellow. Let us self-intercross the F1 genotype to obtain the second filial generation, parental phenotypes. Plants with round and yellow seeds crossed with plants that have round and yellow seeds. Parental genotypes. Heterozygotes with heterozygotes from F1 generation. Meiosis leads to the formation of gametes. The principle of independent assortment works here, as we discussed in F1 generation crossing, to form the gametes. Any one of a pair of characteristics may combine with either one of another pair. When this occurs, we form these gametes. It also occurs on this side. Random fertilization then forms the F2 genotypes. Since we have many gametes, we are going to use a Punnett square. We have the gametes above. Random fertilization. This is the square, which we then insert in the gametes. It also occurs above. Crossing the gametes. Starting with this, we get such a combination. We repeat the same procedure. We continue to fill the whole table, and we come up with such combinations. Let's identify the physical appearance. Remember, capital R is round, which is dominant over the small r, wrinkled. Yellow capital Y, which is dominant over the green small r. This one is round yellow, followed by others. These are 9. This one is round green, plus more 2, this makes it 3. Then we go to wrinkled yellow, the other 2 making it 3. Wrinkled green is 1. This gives a ratio of 9 to 3 to 3 to 1. The 9 round yellow and the 1 wrinkled green are parental types. They resemble parents we started with to form the F1 generation. The 3 round green and 3 wrinkled yellow are the new combinations, called recombinants. These form when there is independent assortment and segregation of genes on the chromosomes. This ratio forms Mendel's dihybrid ratio, complete dominance. In this lesson, we have seen the second law of genetics, known as the principle of independent assortment, and the 9 to 3 to 3 to 1 ratio therefore. The two pairs of characteristics, seed shape and color, whilst combined in the F1 generation, separate and behave independently from one another in subsequent generations. This forms the basis of Mendel's second law, or the principle of independent assortment, which states that any one of a pair of characteristics may combine with either one of another pair. Leave a comment. Let us continue to find out the test cross ratio. Do not forget to leave a comment and a question. In a test cross, we use heterozygotes with double recessive parental genotypes, plants with round and yellow seeds crossed with plants that have wrinkled and green seeds. Parental genotypes, heterozygous crossed with double recessive. Meiosis leads to the formation of gametes. From here, we get these gametes, with the principle of independent assortment. As we discussed, this also occurs on this side. However, since all the gametes on this side are the same, we simply write all. This is followed by random fertilization. Still, we are going to use a Punnett square, leave a comment. The Punnett square, gametes. We cross the gametes to the following combination. These are the offspring genotypes. Physical appearance: 1 round yellow, 1 round green, 1 wrinkled yellow, 1 wrinkled green. This forms the 1:1 to 1:1 ratio. This applies to Mendel's dihybrid inheritance, complete dominance, independent assortment, leave comment. Summary of the mindless dihybrid inheritance ratio, complete dominance. The F2 phenotype ratio is 9:3 to 3:1. The test cross ratio is 1:1 to 1:1. There is independent assortment and segregation. During metaphase 1, homologous chromosomes line up side by side on the spindle before separating. Different pairs of homologous chromosomes behave independently of each other. The way one pair of homologous chromosomes arranges themselves on the spindle and subsequently separate does not affect the behavior of any other pair of chromosomes. Each characteristic of an organism is controlled by a pair of alleles, 2 alleles for each character. If an organism has 2 unlike alleles for a given characteristic, one may be expressed to the total exclusion of the other. One allele is dominant, another is recessive. During meiosis, each pair of alleles separates, segregates, and each gamete receives one of each pair of alleles, the principle of segregation. During gamete formation in each sex, either one of a pair of alleles may enter the same gamete cell and combine randomly with either one of another pair. Second law, the principle of independent assortment. Each allele is transmitted from generation to generation as a discrete, unchanging unit. No mutations occur. Each organism inherits 1 allele for each characteristic from each parent. Summary of similarities between events occurring during meiosis and fertilization and Mendel's hypothesis: factors are transmitted from generation to generation as discrete units. Factors are transmitted from generation to generation as discrete units. Next, each organism inherits 1 factor from each parent. Homologous pairs of chromosomes are restored at fertilization, each gamete contributing 1 homologous chromosome. Characteristics are controlled by pairs of factors. Diploid cells contain pairs of chromosomes, homologous chromosome. Pairs of factors separate during gamete formation. Homologous chromosomes separate during meiosis. Each gamete receives 1 factor. 1 homologous chromosome passes into each gamete. Let me hope you have gained something out of this lesson, free to share it with your friends. Carry out this question. Find the F2 ratio and the test cross ratio. Thanks for joining me today. Don't forget to subscribe and hit the notification bell so you never miss an update. Dive into our other lessons to continue expanding your biology knowledge. See you soon.